Hello, Namaste. I am JV'n. I am Dr. Anupama Goyal. I am working as Associate Professor in Botany in Jyoti Vidyapeeth Women's University, Jaipur. Today I will deliver my lecture on Chara, which is an important algae, and this Chara has been studied by undergraduate and postgraduate students. So now I will first tell you about I'm associate professor in botany. I will tell you about the classification of Chara. Well, Chara has been placed in division Charophyta, and the class of Chara is Charophyceae, or we can say Chlorophyceae. Class is Chlorophyceae, order is Charales, family is Characeae, and the genus is Chara. Now I talk about the occurrence of Chara. As you see, Chara is represented by 188 species. Chara is freshwater green alga, and this green alga, it is found submerged in shallow water ponds, tanks, lakes, and slow running water. So Chara baltica is one of the species which is found to be grown in brackish water. And now Chara fragilis is found in hot springs. Next, Chara is found in hard fresh water which is rich in organic matter, calcium, and deficient in oxygen. Chara plants are often encrusted with calcium carbonate, and so these are called as stone words. So the question is, why Chara algae is also called as stonewort? Because these plants of Chara, they have the encrustation of calcium carbonate. Chara emits disagreeable onion-like odor due to the presence of sulfur compounds, and Chara heti grows trailing on the soil, and Chara nuda is found on mountains. So Chara is represented by about 30 species, of which common Indian species are Chara jaylanica, Chara brownie, Chara gracilis, Chara heti, and Chara segment, and Chara. This is the thallus of Chara. This diagram shows the thallus of Chara, and in this diagram you can very well see the main axis, thallus showing the main axis, and from the main axis, this main axis is divided into nodes and internodes. This main axis in Chara is divided into nodes and internodes, and from the node the branches are coming out. So here in this diagram you can see there are two types of species of Chara, monoecious species and the dioecious species. Now I tell you about the vegetative structure of Chara. I tell you about the vegetative structure of Chara. The thallus of Chara is branched and multicellular, and it is macroscopic. It means macroscopic means it is large in size, and thallus is normally 20 to 30 centimeter in height, and often it may be up to 90 centimeter to 1 meter. Some species of Chara like Chara heti are small, and they may be up to 2 to 3 centimeter long. So the plants in appearance resemble equisetum. So Chara is also commonly called as aquatic horsetail. The thallus of Chara is differentiated into rhizoids and the main axis. And when we see the cell structure of Chara, we see the Chara cytoplasm shows the streaming movement. So here in this diagram, which shows the cell structure of Chara, you see in this outermost layer is the cell wall. This is the cell wall, and inside this there is the cytoplasm or the protoplasm, it is divided into plasma cell and plasma soul, and there are a number of nuclei which are scattered in the cytoplasm, and in the center you see the vacuole, the central vacuole. So in the center of the cell, as you see in the diagram, there is a central vacuole, and in the periphery there are a number of nuclei, and even the starch grains are found in the cytoplasm. And this vacuole membrane is surrounded by tonoplast, means the membrane of the vacuole is called as tonoplast. And as you see in this diagram, the cytoplasm shows streaming movement. And now I talk about the cell structure in Cara. Well, you have seen the diagram. The nodal cells are smaller in size and isodiametric. The cells have dense cytoplasmic uninucleate. The cells are dense cytoplasmic uninucleate with few small ellipsoidal chloroplasts, and the central vacuole is not developed. Instead, many small vacuoles are present, and the cytoplasm is differentiated into outer exoplasm and inner endoplasm. So the internodal cells are much elongated. The cytoplasm is present and around the cytoplasm is present around a large central vacuole. So this is a diagram which shows there is a large central vacuole, and around it the cytoplasm is present. The cells are multinucleate and contain many discoid chloroplasts. The cytoplasm is divided into exoplasm and inner endoplasm. The endoplasm shows streaming movements. The cell walls between the nodal cells and the internodal cells are porous, which help in cytoplasmic continuity between the cells. Now I talk about the rhizoids. In the rhizoids are white thread-like and multicellular uniseriate and branched structures, and the rhizoids arise from the rhizoidal plates which are formed at the base of the main axis or from peripheral cells of the lower nodes. The rhizoids are characterized by the presence of oblique septa. So one of the most characteristic feature of the rhizoids of Cara is that there is a presence of oblique septa, and the rhizoids show apical growth, and these rhizoids help in the attachment of the thallus of Cara to the substratum, that is mud or sand, and these rhizoids also help in the absorption of minerals and in vegetative multiplication of the plants by forming bulbils and secondary protonema. Now I talk about the main axis of Cara. Well, main axis is erect, it is long, branched, and this main axis is differentiated or divided into nodes and internodes. The internode consists of single elongated or oblong cell, and the internodal cells in some species may be surrounded by one cell thick layer called cortex, and such species are called corticate species. Such species are called as corticate species. The species in which the cortical layer is absent are called ecorticate species. Means what are corticate species? The species of Cara in which cortical layer is present are called cortical species, and the species of Cara in which the cortical layer is absent or lacking are called the ecorticate species. The node consists of a pair of central small cells surrounded by 6 to 20 peripheral cells, and the central cells and peripheral cells arise from a single nodal initial cell. On nodes develop four types of appendages. First, branches of limited growth. Second, branches of unlimited growth. So first of all, I will talk about the branches of limited growth, which are present on the nodes of the main axis in CHARA. So the branches of limited growth arise in whorls of 6 to 20 from peripheral cells of the nodes of main axis or in branches of unlimited growth. These are also called as branchlets, branches of first order, primary laterals or leaves. So the students, what is the other name of the branches of limited growth? These are also known as branchlets. Branches of first order or primary laterals. These branches stop to grow after forming 5 to 15 nodes, and so they are called branches of limited growth. And the stipulodes and reproductive structures are formed on the node of these branches. Now I will talk about the branches of unlimited growth. So they arise from the axils of the branches of limited growth. So these are called as auxiliary branches or long laterals. So the branches of unlimited growth are also called as auxiliary branches or long laterals. These are divided into nodes and internodes. At nodes, they bear primary laterals, and these branches look like the main axis. Their growth is also unlimited like the main axis. Next, the stipulodes. So what are stipulodes? The basal node of the branches of limited growth develops short, unicellular, oval and pointed single cell outgrowths, and which are called as stipulodes. In most of the species of CHARA, for example, CHARA burmanica, the number of stipulodes at each node is twice the number of primary laterals. Such species are called as bistipulate. Now in simple terms, students, what do you understand by the term bistipulate? Means when there are only two stipulodes, then we call them as bistipulate. Depending upon the arrangement of stipulodes, CHARA can be divided into haplostephanous, in which the stipules are arranged in a single row, and for example in CHARA brownie, and diplostephanous means the stipules are arranged in two rows. One of the best example is CHARA delicatula, means when the stipules are arranged in two rows, it is called as diplostephanous condition. Now I talk about the reproduction in CHARA. Reproduction in CHARA takes place by vegetative and sexual methods, while the asexual reproduction is absent in CHARA. So vegetative reproduction takes place by the following methods. So how the vegetative reproduction occurs in CHARA? It takes place by the formation of bulbils, by the formation of amylum stars, and by the formation of amorphous bulbils. So first of all, now let me talk about the vegetative reproduction in CHARA by means of bulbils. What are bulbils? The bulbils are spherical or oval tube-like structures which develop on rhizoids. Bulbils are formed or found in CHARA, aspora or on the lower nodes of the main axis. For example, in Chara baltica also bulbils are found. The bulbils on detachment from the plants germinate into new thallus. Now next is the amylum stars. In some species of Chara, as for example Chara stelligna, on the lower nodes of the main axis develop multicellular star-shaped aggregates of cells. These cells are full of amylum starch, and so they are called as amylum stars. So what are amylum stars? These are multicellular starch-shaped aggregates which are filled with amylum stars. So the name is amylum stars. The amylum stars on separation or on detachment from the plants develop into new Chara thalli. Now the amorphous bulbils, these are groups of many cells which are irregular in shape and which develop on the lower node of the main axis. For example, Chara delicatula, or on rhizoids, for example Chara frezifera and Chara baltica. The amorphous bulbils are perinating structures. When the main plant dies under unfavorable conditions, and these bulbils survive and make the Chara plants on return of favorable conditions. So now I show you the vegetative propagation in Chara, and first diagram shows the stem bulbils. These bulbils, by means of bulbils, vegetative reproduction occurs in Chara, and the B diagram shows the root bulbil, and the C diagram shows the amorphous bulbils, and the D diagram shows the amylum stars. See, these are amylum stars, and by means of which the Chara reproduces vegetatively. Now I talk about the sexual reproduction in Chara. So now the sexual reproduction in Chara is highly advanced oogamous type. The sex organs are macroscopic and complex in organization. The male sex organ in Chara is called as antheridium or globule, and the female sex organ is called as oogonium or nucleus. Most of the Chara species are homothallic. The male and the female sex organs are born on the same nodes. For example, in Chara jaylanica. Some species of Chara, like Chara valishae, are heterothallic. So what do you understand by the term heterothallic? The male and the female sex organs are born on different plants. The sex organs arise on the branches of limited growth or on primary laterals. The nucleus lies above the globule. So thank you. We will continue